Uh, Thank you so much for the nice introduction, and thank you so much for the kind invitation. Actually, this is my second time visiting U Michigan. I remember about 2 years ago, I was presenting here, invited by Professor Peter Song on causal mediation. The title I still remember is Deep Learning to Causal Mediation. Actually, I'm talking about the mediation. I'm kind of indirectly connected to U Michigan through a mediator, which is Professor Xihong Lin. U Michigan, Xihong Lin, me. So Xihong served as a mediator between me and U Michigan. It's truly my honor to be here. I know the selection process is quite competitive, so that gives me some pressure. I will try my best to do the job. So today, my talk, as you can see from the title, is a semiparametric causal inference for right-censored outcomes with many weak invalid instruments. Actually, yesterday night, I modified my slides to include a few slides on AI. I think that's probably interesting to some of you. So to motivate this talk, I've been working on UK Biobank data for multiple years. Recently, there's a paper by Ronek Day, which is a former graduate from U Michigan Biostat. There's a paper in Nature Communication. They look at all the time to event outcomes in UK Biobank, ascertained from electronic health records. As you can see from this figure, this is the medical records. So the time to event or survival time is defined as the duration from birth or baseline recruitment to the earliest date of diagnosis for the disease of interest, obtained again from EHR, electronic health records, or registry data, which you can find here from UK Biobank. And the censoring time, defined as the time from birth to the last available follow-up, the most recent clinical visit, record entry, or death date, if unrelated to the disease, whichever occurred last. Here, just to define time to event and the censoring time. Well, the scientific question of interest is, this is one example, does a higher body mass index or BMI accelerate the disease onset time? So that's the whole scientific question we want to answer. Here's the data structure, very typical data structure. Suppose we have 4 subjects. The first 2, they have observed event time. Subjects 3 and 4, they have censored survival time. So we use T1 tilde, T2 tilde to denote the observed event time, and the C3, C4 tilde to denote censoring time. So the question is, if I increase my body mass index, actually I did in the past 2 years, by 1 unit, will the disease onset time will be accelerated? It occurs earlier. So I have to introduce some notation to proceed. Let's T tilde and the C tilde be the true survival time and the censoring time. And the Y tilde is the minimum of the 2, will be the observed survival time. And the delta is the indicator of whether the outcome is uncensored. And we use T is log T tilde, C the log C tilde, Y is log Y tilde, just on a log scale. And X to denote D-dimensional observed baseline covariates. Can be age, can be sex, can be other covariates you want to include in your model. And A is exposure. In this example, A is just BMI. Z, Z1 to M, denotes an M-dimensional vector of genetic variance. So now the genetic data will be included. And some notation here, O denote all the data, Y, delta, A, ZX. OT is TAZX, you remove Y, OA is AZX. So that's the notation. I bet all of you know this model, so I don't have to go to the detail. It's called accelerated failure time model, AFT. But here, the new thing is we have unmeasured confounder. So the question is, can we run the standard AFT regression to obtain the causal effects beta 0? So we want to know beta 0. A is BMI, T is log survival time, X is covariance, U is unmeasured confounder, can be any lifestyle, diet factors. And epsilon had a conditional mean 0. So the question, the statistical question is, or the causal question is, how to identify causal effects beta in the presence of unmeasured confounder U. U, we don't know U. If you run a regression here, we don't know U, so the beta 0 cannot be recovered. We cannot run regression here because of unmeasured U. So that motivates the causal method called instrumental variable. We need additional variable to identify the causal effects. This variable we use is called instrumental variable. We need some assumptions of what kind of data can be used as instrumental variable or IV. So we can use human genotype or genetic variance. So Z here denote genetic variance, like SNPs. A is BMI, Y is time to event. U unmeasured confounder. The interest here is to recover and estimate the causal effects of beta 0 of A and Y. We need 3 core assumptions that define a valid IV. So the first assumption says Z must be correlated with A. Z is correlated with A. This correlation is just correlation, does not has to be causation. Second one is U cannot influence Z. So there's no arrow from U to Z. This assumption is essentially says Z is randomized. Z is randomized. The third assumption says exclusion restriction, which says Z affects Y only from A. It's called exclusion restriction. It's a very strong assumption, by the way. So if you use genetic variance as instrumental variable IV for causal inference, this particular type of IV methods is called Mendelian randomization. The reason is Z here is randomized according to Munder's law of inheritance. That's why we call it Mendelian randomization. So 2 words. This is Munder, from biology, high school biology. We probably already know who is Munder. So Mendelian randomization is very much like RCT, randomized control trials. As you can see from the structure, you have parents due to random assortment of genes according to Munder's law. You have offspring here. The genotype ideally is randomized. So you look at the people with mutant allele versus people with Y-type allele. You compare the results. That's a very typical standard Mendelian randomization. If you have this, everything is straightforward, ideally. But in practice, things are not so ideal. We have challenges. One particular challenge is a biological phenomenon called pleiotropy. What is pleiotropy? You look at this figure here, one single gene here, this red segment. This is called beta globin gene mutation. Actually, it's called single cell anemia. This single gene actually is related to multiple traits of human diseases. This is called pleiotropy. One gene, multiple traits, pleiotropy. If you have pleiotropy, you have trouble. The trouble is the exclusion restriction assumption will be violated. And there are many papers talking about pleiotropy. By the way, pleiotropy can be an interesting problem in genetics, but here is something we don't want. It's a trouble. So it depends on your research question. It can be something interesting, but it can be something uninteresting depending on the setting, okay? So we need some mathematical models or statistical models. So when you have a violation, so a violation of exclusion restriction, the structural model becomes this. T beta 0A plus this green term. Green term is a function of Z and X plus some xu. This is u is unknown plus epsilon. What is green term? The green term is here. It's a direct effect of z on t, not through a. So this is pleiotropy. A genetic variance affects disease onset time, not through BMI, through other biological pathway. If you have this green arrow, you have this green term. If this green term alpha is 0, this arrow is gone, back to the standard setting. So this alpha term is really the trouble. Now we try to handle this alpha term. We call this alpha term violation bias term, okay? Because it violates exclusion restriction. And we also have a structural model for the exposure BMI. This is standard. We don't have to worry about this part. We worry about alpha, this violation. So we try to figure out how do we recover beta 0 when this alpha is present. There's some other challenges. For example, Z to A, this genetic association is weak, but this is testable. This green term is not testable. You can never test it using any empirical data. So the challenge 1 is U is unobserved. Challenge 2 is this alpha green term, this violation due to pleiotropy is unknown. So you have 2 challenges. And how do we address the challenges? That's the whole difficulty, the question. So this is called identification. In causal inference, the first step is to identify, can we identify or can we learn the causal parameter using empirical data? This is called identification. Now, the idea is, can we construct a function h? This h is a function of z, x, and a such that h function multiplied by t minus beta 0, take expectation, this is 0. If we can construct h function, we plug in, this is 0, we can solve for beta 0. This is unbiased estimating equation. In econ, people call it moment condition. In our field, we call it estimating equation. Different names, the same thing, okay? So here's our choice. We construct this h function as this. The first one is a residualized instrument, RZ. Z minus expectation of z conditional x. So this is residual of z. Second one is residual of BMI. You regress BMI on z and x, you take the residual. Why we like a residual? From basic regression, whatever cause. Residual is orthogonal. Here's a residual. This residual is orthogonal to the space you project to. We need something that is orthogonal. If it's orthogonal, it will be 0. You got the idea? So this term, residualized exposure, BMI, this is orthogonal to any function of Zx. This is the key idea. A minus, you project A to Zx. This residual is orthogonal to any function of Zx, including alpha Zx. Whatever alpha is orthogonal. So you take expectation, it's 0. We solve the equation, we're done. So this is the idea. We construct this term h, then we cancel the duality effect term, which is a violation term. And we use some other trivial assumptions. The whole equation here, this equation will be 0. If it's 0, we can solve for beta 0. So the causal effect is recovered. That's our identification strategy. Actually, we have a closed form solution because this is linear in the causal effects beta 0. So here is the beta 0 identification formula, HT, HA, on average ratio. This is very much like the traditional IV identification formula. What you have changed is H. In the traditional setting is Z, now it's H. You construct something that make it work. To make sure this is identifiable, the denominator cannot be 0. That's the testable assumption. The denominator cannot be 0. There's one challenge here. What if the denominator is on 0, but very small? A tiny number in the denominator. Do you want a number very small in the denominator? No. But what if it's small? If it's small, we have a so-called weak identification problem, and we will address that. So this cannot be 0, and we do some algebra. The denominator cannot be 0, and after a few lines algebra, maybe ChatGPT can help you to do the algebra, and you show the variance of A conditional on Zx does depend on Z. So this is the BMI must be heteroscedastic in the genetic variance. Okay, that's the condition. It's a testable condition. So the identification relies on heteroscedasticity of the exposure variable with respect to the genetic IV. Is this real in genetics, in biology? You don't want to make up some weird identification never exists in nature. Well, it's real. I can show you some evidence. You can see this is a genetic study. When you have a C allele versus a G allele, this is the distribution of BMI. This purple term, this purple distribution is the C allele. People with the C allele, people with the G allele. So you can see the distribution, they are different in terms of variance, not just mean. Here shows you another example. So genetic variance can change the variance of BMI. Show some other examples here. This is published in Nature. FTO genotype is associated with the phenotypic variability of body mass index. Also, a lot of evidence in UK Biobank. This is not Z genetic variance change the mean, it changed the variance. Okay, so we call such kind of genetic variance that change the variance of a trait is variance QTL or VQTL, quantitative trait loci. So what are the reasons that contribute to such phenotypic variability across the genotype? Gene by environment and gene by gene interactions contribute to the phenotypic variability. As long as you have gene environment, gene by gene interactions, you will observe phenotypic variability. You can prove that. So when you have a right censoring, how do we identify this? The approach is we use augmented inverse probability of censoring weight, or AIPCW, to construct a new moment function, psi. This is psi. So G is an influence function without a censoring. And this G0 here, this is the distribution of Y conditional on OA. So this is a conditional distribution. Delta is 1, we will upweight this subject. Delta is 0, we use some kind of imputation. Very much like a missing data, but slightly different. So we impute the unobserved survival time when delta is 0. So this weight here, G0Y, is defined as the probability of a censoring time larger than Y, refers to a conditional distribution of the log censoring time. And the C0, the imputation here, I say imputation is in high-level imputation. The imputation is the conditional expectation of uncensored influence function conditional on observed data OA. It's a conditional expectation of uncensored influence function given observed data. So that's what I mean by imputation. So here is a cartoon to illustrate the idea. Again, we have 4 people here. Person 1, person 2, smaller weight, larger weight, you live longer, you have larger weight, okay? And subject 3 and 4, they are censored. So we predict the censor time, we predict the censor time. And under non-informative censoring, all the subjects will contribute to the estimation. So here we show a closed form solution for beta 0, the causal effects on the right censoring. This is the formula. So to estimate beta 0 with any finite sample, we just need to estimate all the nuisance functions in this formula. We plug in all the nuisance function estimation, we can estimate the beta 0. So there are many nuisance here. Nuisance 1, bunch of nuisance. How do we estimate the nuisance? The nuisance function can be highly nonlinear, complicated. We never know the true nuisance. So that's the estimation challenge. There's one special nuisance, which is the conditional distribution of the censoring time, G. I put it in the very end because it's a special one. I'll tell you why it's special. So all the nuisance here can be divided into two groups. One group, those nuisances satisfy the Neyman orthogonality condition. The other group, the nuisance does not satisfy Neyman orthogonality condition. So they are non-Neyman orthogonal. What is definition of Neyman orthogonality condition? It's the estimating function. You take a functional derivative with that nuisance. If it's 0 at the truth, that nuisance is Neyman orthogonal. So this is your estimating function. This is your nuisance. They are orthogonal. If you move around, up and down, this is flat, no change. What if it's not orthogonal? There's angle. Go down, this one will be changed. That introduce bias. You see that? So that's why we need this Neyman orthogonality condition. But unfortunately, this conditional censoring distribution G, which is used to upweight observed people, this is non-Neyman orthogonal. You take derivative, it's not 0. And this is the first time probably in the literature we formally address this challenge. So all the double machine learning papers depends on Neyman orthogonality condition. If Neyman orthogonality condition does not hold, you don't have double machine learning. So you assume this. But now we have a non-Neyman orthogonal nuisance. This is a challenge for semiparametric inference. Okay, so we'll address this. How do we do this? Here's a table to summarize Neyman orthogonal versus non-Neyman orthogonal nuisance. If you have Neyman orthogonal, life is easier. If you have non-Neyman orthogonal, you need more efforts to make it work. As you can see, definition, derivative is 0, pathwise derivative is not 0. If you have Neyman orthogonal nuisance, we have weaker rate on estimation. It's only 1/4 rate is sufficient to ensure our causal effects estimation is okay. But if it's non-Neyman orthogonal, it's much more complicated. In terms of first order bias of the causal effects estimator, if it's Neyman orthogonal, it's automatically eliminated by the condition. If it's non-Neyman orthogonal, there's no guarantee. We need to construct a very smart estimator to kill the bias. The variance is the key part. If Neyman orthogonal, the variance of a beta height estimator, no change. If it's non-Neyman orthogonal, there's a price to pay. You have a new variance component you can never avoid. That's the price we have to pay. Okay, another challenge is weak identification. When this denominator is small, we have weak VQTLs, lead to small denominator. So this is small, that's not good. So our solution is to combine many weak VQTLs to strengthen identification. If you have many VQTLs, there's two benefits. One benefit is you can increase efficiency. Second one, it permits over-identification test for model diagnosis. How do we combine many weak moments? There are basically two approaches. One is generalized method of moment. The other one is generalized empirical likelihood. So here is empirical likelihood. In the paper, we consider 3 different methods: ET, exponential tilting, empirical likelihood, and continuous updating. They have different format, functional form for rho. They have different theoretical properties. So high level, generalized empirical likelihood can be used to combine many weak moments to make the estimation more accurate. Here's a graphical comparison of the 3 different generalized empirical likelihood. The blue one is the COE, the red one is the ET, and the green one is EL. Here we show the first order derivative, here the second order derivative. And this is related to the curvature. So the variance will be slightly different stability. Estimation procedure, we first estimate the non-Neyman orthogonal nuisance using a local Kaplan-Meier estimator. For the Neyman orthogonal nuisance, we use flexible deep neural networks to learn conditional expectations. And then we plug in all the estimated nuisance function into the GEL loss function so we can get an estimate of the causal effects beta 0. Here's a new type of asymptotics, so-called many weak moment asymptotics. I won't go to detail, but I can show you two figures here. What do I mean by weak moments, strong moments? Strong moments is the conversion rate is root n for beta estimation. If the rate is slower than root n, it's called weak moments. What do I mean by many moments? It means the number of m is allowed to diverge. Fixed moments, m is fixed. So talking about deep neural networks, we need fast enough conversion rate for deep neural networks to ensure valid inference for our causal effects estimator. So the rate we derived is this eta hat is a deep learning estimator. Eta 0 is the true conditional expectation, the nuisance. Must satisfy this rate to ensure valid inference for our causal effects estimator. You can ask, is this rate achievable? Yes. For certain smooth function, eta 0, and a certain architecture of deep learning, it is achievable. And one advantage is we don't need cross-fitting. Unlike double machine learning, cross-fitting is not necessary. We can use some kind of empirical process technique to justify the convergence. Yes?
So to clarify, what is M here?
M is the number of SNPs, number of IVs.
Okay, go back to the previous slide.
Yes.
So can you explain again what is the many moments here?
Many means the number of IVs diverge, but at a slower rate compared to sample size. Diverge, but not highly massive, slowly goes up.
And here is the actual convergence rate.
Yes.
Okay.
Mu A is typically called a concentration parameter. It's too technical, but I can discuss later. So high level, we need some smooth function for the conditional expectation. We need some kind of architecture as deep neural nets, so we can achieve this. If we achieve this, we can have convergence for the causal effects estimator. So here is the main result: consistency and asymptotic normality. Our GEL estimator is consistent, and this is the mu and the rate, as you said. So this mu n is no longer root n convergence. It's slower. Why is it slower? Because it's weak. If the moment is weak, you cannot walk fast. Then it's slower. That's the intuition. If it's strong, it converges faster. So we have three variance components here: H, V1, V2. This H is the information matrix from moment conditions. Very traditional result. V1 is the variance from many weak moments. We know this from literature. V2 is new in this paper. V2 is the variance from estimating non-Neyman orthogonal nuisance. That's the price to pay for V2. How do we measure weak identification? We use this approach. You compute the residuals from BMI model. You test the variance dependence on instrument by fitting this linear regression. And you can calculate the F statistic from this model. If F statistic is bigger than 2, good. Smaller than 2, weak. That's how we do this in practice. The 2 is based on empirical simulation. And when you have multiple IVs, actually that allows for over-identification test with the censored data. This is a test we propose. It's different from traditional test. This is a generalization of the Sagan-Hansen test because we have a new term here, V3. This V3, again, is because of we need to estimate non-Neyman orthogonal nuisance. That's the price to pay, again, for this test. So the power is slightly lower when you have non-Neyman orthogonal nuisance. For UK Biobank data, the original data set is about 500,000. In this application, we focus on senior adults. Age is bigger than 65 with EHR data. The time event we have defined from baseline recruitment to disease diagnosis time. Right-Censored. Exposure is BMI, covariates, age, sex, and systolic blood pressure. Instruments, we have 93 instruments, a decent number, 93. So M is 93. They are all variance QTLs for BMI. So look at the results. So time to event outcomes are certain from UK Biobank EHR. Those are the final code outcomes, events, sample size, sensory rate. Okay? From ICD code in the medical records. So those are the analysis results. We compare with the benchmark, the traditional AFT model with measured covariates. Yes?
I have a question for the event and the censoring. So if someone doesn't have the disease, is there ever an event where it says definitively that they don't have it?
Sorry, can you repeat?
Oh, sorry. I was just saying for the events and censoring for those diseases. So if someone doesn't have a chronic disease, is that always considered censored, or is there some point at which it's observed that someone doesn't have that disease indefinitely?
That's a good question. Honestly, we just use the code from the Nature Communication. We run the code and get all the data from the medical records.
Gotcha.
I think your question is very important, but I don't have an answer for your question.
Thank
Yeah,
you.
this is how we process the EHR and extract the censoring time and the event time from UK Biobank following the Nature Communication paper. As I said, is from the first author is a graduate from U Michigan Biostat. But we can talk about the details later, how we extract the data. So the model we propose is called the MAVE Survive. This is our method. We compare with two traditional AFT model without covariates with covariates, observed covariates. Those are the effect size estimation. P-value, weak IV diagnosis is all bigger than 2, and the over-identification test. The tests are not significant, which are good. We don't want them to be significant. If it's significant, we are in trouble. So they are not significant, that means our model specification okay. No evidence against our model specification. In this setting, we don't want them to be significant. Well, we need to be honest, but they are not significant. So the p-values here shows you the association between BMI and the time to event. So those are the effect size. Next step, how do we interpret those effect size? The first one is ischemic heart disease, diabetes, and dementia. Causal effects here, interpretation here. The interpretation is interesting. With 1 unit increase in BMI, 12% early onset for ischemic heart disease. Just 1 unit from 25 to 26. The disease onset time will occur earlier. So that's bad. So BMI is bad for the heart disease. For type 2 for diabetes, the effect size is this. 1 unit increase in BMI, 27% earlier disease onset time. Very bad too. BMI is critical for type 2 diabetes. The third one is a little bit controversial. 1 unit increase in BMI, 8.7% delayed onset. In the literature, this is called obesity paradox. So among senior people above 65, higher BMI are bad for ischemic heart disease, bad for diabetes, seems to be protective for dementia. Now this is a decision. Should increase BMI or should you reduce BMI? It's bad for these two. It seems to be good for these two. This is called obesity paradox. If you compare our methods, we serve, actually we correct attenuation bias of standard AFT model. Standard AFT model tend to give you smaller effect size because of unmeasured confounding. Our effect size are bigger because we correct unmeasured confounding. So that's the results from this UK Biobank. But I have to say, there are always caveats. The data is not perfect. We address confounding bias, but we don't address other types of bias. There could be measurement error. There could be misclassification. There could be a selection bias. The different kind of bias will cause those, paradox. So I don't claim this is scientifically 100% correct, but it provides some evidence. To conclude, we propose identification strategy on the right censoring with invalid instruments. And we propose this semiparametric gel inference in the presence of a non-Neyman orthogonal nuisance. This part is new in the semiparametric literature. And we figure out how to do valid inference with many. Many means the number of instruments diverge. Weak moments. Weak means the rate is not root n. That's the definition. And we also propose how to measure weak identification and how to do over identification test for model diagnosis purpose. And we have a Python package, which is faster than R, as we discussed during lunch. You need to learn Python. So Python can do faster computation for biobank level data. And I want to thank co-authors, some students. I don't have students at Columbia, so I still use students in Hong Kong. So those are the students in Hong Kong and the collaborators in Hong Kong. I want to thank UK Biobank for this application. And this is a QR code. If you scan it, you will be surprised. So I will share the slides. It's not done yet. I still have 20 minutes. I will spend 10 minutes on AI. I was advised yesterday, you better talk about AI. Okay, so I spent a few hours in the hotel yesterday by adding some new slides. So let's see. It's not a perfect slides, but probably can serve the purpose. So it's called a generative AI in drug discovery and repurposing for Alzheimer's disease. That's actually my focus for my R1 project, so I have to do it. So let's look at the normal versus Alzheimer's disease brain. On the left is the normal brain. On the right is Alzheimer's disease brain. What you can see here, I highlight here. For Alzheimer's disease patient's brain, you observe amyloid beta plaques. This is the plaques. If you do PET scan or postmortem, you cut the brain, you can see that. But postmortem, of course. Before they die, you can do PET scan, you observe this. Amyloid beta plaques in the human brain for every Alzheimer's disease patient. The question is, what's going on here? We look at the protein level data. For a protein is basic functional units in human body. A protein is simply a sequence, a linear chain of amino acids. That's it. Here is the amino acid sequence. But due to chemical properties, those amino acids will fold into a complex structure. That's called a protein folding problem in biology. It's actually a big problem in biology for many years. In the past, people use experimental techniques, X-ray or some fancy equipment to understand the structure of a protein after folding. Last year, Google DeepMind developed this AlphaFold 3, which they got the Nobel Prize in the same year in October. So this is a generative AI tool for protein structure prediction. Here is how they work. Protein and this. That's AI. So we have this paper published about 10 months ago. We use this tool for Alzheimer's disease research, combined with causal inference. So causal inference, AI, together to solve Alzheimer's disease problem. So this integration of AlphaFold with causal inference. Here is the basic pipeline for this research. We first select genetic variants, and we do some kind of causal inference. We found some proteins, and we look at the structural changes due to genetic mutations, so-called missense mutations. Missense mutations are mutations that will change your amino acid, will be a different amino acid. That's called missense mutations. And then we look at, this is the Y-type protein without a mutation. This is the mutated protein. The protein is different. The structure is different. And then recently, we improved our causal inference methods. We found more proteins here. For example, this ApoE TREM2. This is not published yet, probably will publish very soon, hopefully. Those proteins are identified using causal inference methods, using instruments, basically. They are all proteins related to Alzheimer's disease. On the left, those are the bad proteins. On the right, the green ones, they are good proteins. So some protein, actually, they are protective. Some protein, they are harmful. But not all proteins are created equal. Some are bad, some are good. Now, AI again. What we found recently, actually, last month, we used AlphaFold 3 to predict the protein 3D structure alterations. This is a famous protein called ApoE. ApoE epsilon 2, ApoE epsilon 3, and ApoE epsilon 4. If you look at the difference locally, this is a glutamate acid at a residual 127. Residual just means amino acid. Here is cysteine at a residual 130, the local molecules. ApoE2 is a good protein. It's a good variant of the protein. The blue one is a neutral one, and the red one is a better one. If people carry this one, the Alzheimer's disease risk will be much, much higher than these two. If you look at the structure here, we use AI, we predict the distance between them. This is like a 0.26 nanometer, very tiny distance. And this part and this part, they have different charges. One is positive charged, the other one is negative charges. So that cause a salt bridge in chemistry. If you have a salt bridge, this is the first time we identify in the literature using AI. We found this salt bridge between these two due to mutation. What is the consequence of this salt bridge estimated by AI? The consequence is we use another chemistry calculation. It's called buried surface area, or BSA, for the three different proteins. You look at the numbers here. For the good one, the green one, this is number. For the blue one, this is number. And for the red one, this is number. 900, 1500, 2800. So numbers are very different. This buried surface is the protein binding between the three different proteins with amyloid beta protein, which is the bad protein you observe in human brain among Alzheimer's disease patients. because of this binding affinity. Let me explain this binding affinity. So if you have this good protein, this is a binding. That's the surface, that's overlap surface. You have bad protein, boom. Binding is stronger because the binding surface is bigger. That's how what's going on here. And knowing people have bad protein, can we do something? Showing people bad news is not a good way to communicate. You want to give some bad news and you give a solution. So here's a solution. We use this diffusion model. Diffusion model is a forward, backward process, adding noise and denoise so you can learn the structure. Actually, the same for protein structure. So what we did is we use this diffusion model. We de novo design a protein. The blue one is a de novo designed protein. They can bind to the bad protein. The ApoE4 is the red one. The red one is a bad protein for Alzheimer's disease. And we design it using AI, a blue molecule that can bind to this. If it binds to this, it will reduce plaque accumulation in the brain. So can reduce Alzheimer's disease progression and risk. So this one is a drug target. It's an AI designed drug target. Still, in the research stage, in the clinical stage takes longer. But this is how AI can help us design molecules potentially can treat Alzheimer's disease. Can we do this? As I said to the students, can we do this using regression? No. We need some fancy tools, advanced tools to do this. So among all the proteins, actually some protein targets has already been targeted by existing drug for a different disease. For example, this is CD3 protein we identified. This protein has already been targeted by this drug, by Pfizer, for the treatment of acute leukemia. But this protein is also related to Alzheimer's disease. So that provides drug repurposing opportunity up to RCT approval, of course. There's another drug. This red protein is also related to Alzheimer's disease, but this red protein has been targeted by Genentech and Lilly. There are two FDA-approved drugs called RET inhibitors for the treatment of non-small cell lung cancer. So this drug here, the drug here, probably can be repurposed, existing drug. So I want to stop here. Thank you for your attention, and I look forward to any questions and discussion. Thank you so much.
Thank you so much for the excellent talk. We can just go to some questions.
So I have a question about the non-Neyman orthogonal part.
Yes.
So why don't you construct a Neyman orthogonal scar by projecting the influence function to the tangent space of the space of beta? Is it very difficult?
Yes, we tried. We don't like it, but we cannot. We didn't figure out how to do the projection to the tangent space. The tangent space is a little bit hard to characterize. So we
Thank you. And I have another.
but it's possible. Theoretically, it's possible. We don't have a solution.
I have another question about the obesity paradox. So when you do the data analysis, did you consider death or other events as competing risks?
No, we didn't consider. That's why I say there are caveats. Interpretation with caution. Or competing risk is a different problem, actually.
Okay. But you showed the distances between those residues, right?
Yes.
And I'm assuming, are those predicted from AlphaFold?
Yes.
So how do you think about, or do you worry about uncertainty around those predictions?
Yeah, that's a good question. That's about this is really a statistical thinking about AI. AlphaFold, they have their own uncertainty quantification number called PLDDT. That number tells you how certain their prediction is for each amino acid. Actually, it's at atom level. For each atom, it's smaller than amino acid. Compared to lab experiment molecule, it's very accurate.
For the sets of proteins.
Yes.
They have
Yeah.
seen.
Yeah.
Yeah.
So very accurate. I think that's why they got Nobel Prize. So.
Okay.
Yes.
Couple questions. One is BMI treated as time varying?
No, it's yeah, that's a very good question. It's a baseline. Just one-time measurement. Yeah.
And then the second thing is a bit more statistical. So I've often thought about instrumental variables as kind of being a special case of principal stratification under like exclusion restriction, where you have usually a fairly simple kind of instrumental variable, right? Particularly if it's.
That's true.
So I don't know, is anyone giving any thought to that direction for trying to deal with the failure of the exclusion restriction in this setting? I mean, it's a high dimensional instrument, so the whole thing may kind of blow up when you try to plug it into that framework. But
I think that's a very difficult question to explore in your proposed direction. Yeah, so indeed they are related. I think that probably worth exploration how to make it work if we have invalid instruments in the principal stratification approach. But if you use principal stratification approach, the estimate will be slightly different. It depends on which estimate, like in which strata we want to estimate the causal estimate. So the scientific question is slightly different. It's slightly different. So the question to ask which principal strata is the most relevant strata for our public health research. A combination, yes. Thank you. Yes.
This may be a very naive question. How many is too many?
Oh yeah. So many is more like asymptotic sense relative to the sample size. As long as the number of instruments is allowed to diverge, to grow, it's called many.
The more the better?
Not necessarily.
And so how do you know?
How do I know? There are multiple things together. That is really the asymptotic theory part. So there's conditions, technical conditions. So those are the conditions tells you what is M, what is the concentration. So everything together here. Everything put together, if they satisfy those conditions, we can have convergence. Otherwise, we don't have convergence for the causal effects estimator. So there's no a direct answer. It's more involving.
I guess I was, if you go to the next slide, when you have a criteria, maybe next one, criteria for making the.
Here.
Which one is no, I know these conditions, but you have a test, right? If test for which one is the stronger one.
Yes.
Yeah. So, I mean, this is a very like a dichotomized cutoff, right? I mean,
this is empirical guidance.
Right. So if you have many that if you think of this, many are bigger than one, but many are smaller than two, right? But then some are getting smaller and smaller, right? So imagine that you have, it's a continuous kind of set of numbers for all these IVs, right? So Which one do you choose? Which, I mean, obviously you want to keep the twos, but those above the two if with strong identification, right? But with weak identification, you have, lots of ones, maybe even smaller, right? Where do you start? I guess that's.
Oh yeah, question. That's a very practical question.
Right.
Very practical. Yeah. It's more data adaptive approach, I have to say, in practice when you analyze data. Another heuristic we can do is we do the variance testing. So let's go to the data part. Where's the data part? Yeah. So the 93 here, we can do a VQTL testing. We pick the significant ones and we include them in the data. If you ask, is this the best number? We don't know. but we can do standard VQTL like GWAS. But GWAS is on the mean scale. VQTL is on the variance scale of the trait. If we do this, we can select all the VQTLs as people did in the GWAS literature, like these two papers. This is Jian Yang's paper, Nature, and also Jian Yang's paper analyzed UK Biobank. So if they report significant SNPs, and we just include those significant SNPs. That's in practice how we do this. Theoretically, are those the best number? We don't know. Yeah. Yes, Binka.
Since we still have time, do you mind expand on the second part, how statistics help in this effect? I know many of them are kind of mathematics, but you mentioned about causal inference.
Yeah.
That's the critical part there.
You mean in the AR part, right?
Yeah.
Statistics plays a fundamental role. And I have to define our profession. So to work on how do we know which protein is related to Alzheimer's disease? That's the number one question, step one. That's a statistical question. How do we identify proteins related to Alzheimer's disease? That's a causal inference question. So that's what we can do. First step, identify proteins. Second step, AI. So I would say is statistics and AI, they are paired. They're paired together to understand what's going on with Alzheimer's disease. They're not competing. They're more like partners. So statistics is partnered with AI to solve scientific questions. That's how I view statistics in this application. In some other applications, probably people want to use the statistical principles to quantify uncertainty, or sometimes we use AI to improve inference. But in this particular application, they are paired, they are responsible for different parts of the problem. We use statistics, in particular causal inference, to identify causal proteins. Number one. Number two, we use AI to understand molecular level mechanism. Yes.
So following up on that, so if you using the statistics to identify the causal protein,
Yes.
you also use the variant specific results from the genetic data to connect to the AlphaFold?
Yes, that's true. That's what's going on here. So they are kind of combined together. One minute left. Maybe one more last question. I leave tomorrow, so. Students, maybe? Yes.
Why is Python faster than R in that case?
You have to ask who invented Python and who invented R. I think fundamentally they have different mechanisms. Python is more general purpose programming. R was invented by statisticians who are not professional computer scientists. So maybe they have some fundamental computer science level differences. I have no clue. I use them, but I don't design them. So I'm not that expert in computer science.
From my personal experience, R is like closer to more lower levels like C++ and Fortran and stuff.
No. You probably use C++ already, like CPP package behind some numerical calculation. You just use R wrapper function, but when you run it, it's actually C++ is running. But Python is different. Dropbox is written by Python. So Python is much more faster when you work on genetic data, much more faster. Especially for R, you have to load everything into the memory. Python is not. So when you have a huge data set, R will load everything into the CPU memory. You need a huge memory to do this. Python is not. Python read data, process it, release the memory, read data, release the memory. So it's a different architecture. That's all I know. So
This is not really relevant. Have people know, can you call Python from R? You can.
Yes.
It's used to call MATLAB from R.
Yes.
MATLAB.
Yes. You can call
Yes.
from each one call the other one. Yeah. Okay. So thank you again.
Again.